Welcome to the third in a series of special topics in cell isolation webinar. The first one was on mammalian hepatocyte isolation. The second one was on the history behind isolation of human adipose stromal vascular cells, and these recordings can be found on our webpage. This one will be about learnings from developing and applying purified enzyme mixtures for cell isolation. There'll be one poll at the start and two polls at the webinar. These are very short questions. Please, if at all possible, complete the polls. If you have any questions, I ask you to type them in using the chat box, and I'll get to them at the end of the webinar. The webinar will be recorded, and the slides will be shared several days after the webinar. So, without further ado, I will start this presentation. C. histolyticum collagenase-mediated cell isolation. I'm going to turn off my video, by the way, so you can focus on the slides. C. histolyticum collagenase-mediated cell isolations, learning from the development and application of the Liberase HI purified enzyme blend, which was made for human islet isolation. Here we go. Oops. So the outline of the presentation is really what we did learn from Liberase. From these learnings, we developed a useful model for understanding collagenase-protease-mediated cell isolation. And from that model were three really profound insights gained. One, why you have to pre-qualify traditional collagenase products. Insight two is that you can make from an existing formulation, a modified formulation for rodent hepatocyte isolation. And the important thing is from this model, you can develop what I term a rational design model for optimizing collagenase protease enzyme mixtures. So learnings from the development and application of the Liberase product. This is kind of an interesting story. Collagenase P was made in 1990 when I was at Boehringer Mannheim Biochemicals by our German colleagues. And unbeknownst to my German colleagues, this product had a very unique characteristics in that it had very high collagenase activity. Camillo Ricordi, who was at that time at the University of Pittsburgh, found that this was better than any other collagenase on the market. And about 30% of the lots were used successfully to recover high yields of human islets from donor pancreas, which he subsequently transplanted into patients. However, Boehringer Mannheim Biochemicals was in the research business, so immediately became concerned that the product was used to prepare islets were then transplanted back into patients. We sent two people to the University of Pittsburgh to talk to Camillo. And the result of that visit is they invited him to visit Indianapolis to discuss his needs for improved collagenase. This visit, of course, he convinced them that Boehringer Mannheim was a diabetes care company because it sold a lot of glucose strips. So he convinced them that they should organize a project, and consequently 10 people in biochemicals R&D were led to identify, purify, and manufacture the enzymes responsible for human islet isolation. The project was started in January 1993. We identified the components by August of 93, and we launched Liberase HI in November of 94. It took over a year to transfer and scale up the manufacturing process. Just to note, as of a week from today, it will be the 30-year anniversary of the celebration we had in launching this product. So what did we do? We made Liberase HI. As you can see, instead of having all of these contaminating biopigments in the crude collagenase P, which is a brown to black color, we had a white powder. When you compare the two, you can see dramatic differences. We now have defined enzyme activities. 60% C1 is class 1 collagenase, 40% C2 is class 2 collagenase, and we added either thermolysin or dispase to the Liberase product line. Lower endotoxin, highly purified mixture, and it was a defined formulation that could be modified. This product worked for many years, from say 1994 up till around 2002. And at that time, Jonathan Lakey at the University of Alberta in Edmonton received a lot of funding from the JDRF after publication of the Edmonton Protocol, and he started doing biochemical analysis in Edmonton. So we transferred the procedure for an analytical anion exchange chromatographic analysis of purified collagenase to Jonathan. In the panel A, this is what the typical profile, an anion exchange profile was. The numbers indicate the retention time in minutes coming off the column as you add a gradient to elute off the different collagenase forms. And you can see the class 2 collagenase comes off first, followed by the class 1. What Jonathan noticed is he started getting these shoulders or double peaks, as shown in the other slides. And when he did a further study and looked at the lots, you can see that something went awry in the manufacturing. First and foremost, in lot 18, the ratios are reversed. And secondly, you started seeing this back peak, this second peak, C1b peak, we refer to it, in the fractions. And this was very variable. Now, why is this important? Well, he showed, and this is from the data was presented in this, a lot of the data was presented in this publication by Barnett, but he showed in a slide presentation in 2006 at the Islet Cell Research Workshop that in fact, the unsuccessful isolations had a lower, he called collagen degradation activity, he used the Fitzy gelatin substrate and measured the fluorescence. They had a lower, unsuccessful isolations had a lower gelatinase activity than the successful isolations. And you can see the difference where the cutoff of a successful isolation was greater than 300,000 islet equivalents per organ. Of course, this was a significant difference. So how do we explain the result? We had just started VitaCite and immediately got intrigued by a publication in 1998 from the Matsushita group in Japan. And they basically showed that there were three domains in collagenase: a catalytic domain, PKD-like domain, which was initially called a linking domain, and a collagen binding domain. Note that newer knowledge has now split the catalytic domain into an activator and peptidase domain. So there's actually 5 protein domains in both of these enzymes. So the C. histolyticum collagenase is a multi-domain structure. To be functional and cut collagen, you must have a catalytic and collagen binding domain on the same molecule. Non-functional collagenase doesn't have a collagen binding domain. It can't bind to collagen. It can't degrade this macromolecule. So this was all interesting, is how do you interpret, or how do you find out what's really behind the structure? Well, we did a very interesting experiment in 2007. Drew Bridey was really the person who performed the experiment. He's still with us. In that, what we ended up doing was taking the same analytical anion exchange profile, and believe it or not, this lot did work for human islet isolation. And you can see the three peaks in the C1 at 20.1 minutes, the C1b, which we just didn't know exactly what the activity was before we did this experiment, in the C1c. And basically, when we took the peak fractions from each of these four peaks and then put a portion of it into an SDS page to get a mass of the protein, and the other for a collagen degradation activity assay, a CDA assay, on which we had just developed. It was a kinetic fluorescent microtiter plate CDA assay. We found, surprisingly, that intact C1 had the highest activity. As you can see here, in the initial experiment, it was about 100,000 CDA units per milligram purified protein. C2 and the other forms, the C1b and C1c, actually had about a 10% of that activity. We now term the C1b and C1c truncated forms because, as shown in this slide, they had lost the carboxy terminal collagen binding domain. But note, all of these forms are active. These numbers are somewhat different with our newer assay, but the relationship holds. So why is, again, this important? Well, you have to realize most people measure collagenase activity by Wunsch activity, which only measures this, as you can see in the far right side, and the C2 activity, collagenase, the class 2 collagenase activity. It's really important now to look at collagenase as functional being defined as the ability to degrade native collagen. As you can see, intact class 1 has 2 collagen binding domains, just like Matsushita said. Intact class 2 has 1, and the truncated class 1 has lost its carboxy terminal domain, but it's still active. The other forms, if you lose that collagen binding domain, you can't degrade the extracellular matrix or native collagen. So here we're presented with all this new information. And we had decided, well, we might as well we're writing a review on acceptance criteria to accept enzymes for human islet isolation. It was published in 2011, and we decided, well, how can we put this together and make sense of it? And as Albert Einstein said, you don't have to make it complicated, you just have to have a simple model to see if you can resolve and answer any questions. And that's what we did. And the first report of that was in this review we wrote. So the real point that you've got to do to free cells from tissue is overcome the extracellular matrix protease resistance. As we know, and as all of you are aware, cells are tethered to the tissue by cell anchoring proteins. Collagen is the predominant protein, but it really is providing a protease-resistant superstructure that protects and shields ECM proteins from degradation. And this, of course, we know for many years, proteases alone cannot free cells from tissue. Enzyme mixtures must contain a sufficient amount of collagenases, collagen degradation activity to start to free cells from tissue. So what led from Matsushita's initial work was that Josh Saccone and another group in Austria showed that collagenase degrades collagen by synergistic attack by the class 1 and class 2 collagenases. And you can see, and this is a slide from Josh Saccone, that C1 is a process of protease, as he terms it, and it literally shaves off the collagen by moving along a tropocollagen molecule. And you can see what leads the charge, so to speak, is the collagen binding domains, and they pull the catalytic domain behind it. In contrast, C2 is an endoprotease that makes internal cuts on the collagen, as indicated by the saw. And in fact, these two are working in synergy. The cuts probably lead to more binding to the C1 and so forth. But what it really is doing is it's now loosening up that matrix and freeing protease sensitive or exposing protease sensitive sites on the ECM proteins. And here is just some slides that from the Liberase HI product, where we're putting in a porcine pancreas into a Corti chamber. And at that time, we were doing complete digestions of the organ and measuring the islet release using an automated image analysis system, where, of course, people who do islets realize use zinc stains to isolate the islets, because they're the zinc is used to bind together the insulin in the granules. And basically what we see here is that you get complete digestion of that organ. So the collagen is degraded, matrix loosens, and you get release of cells. And you just have to have a sufficient number of these anchoring proteins cut to release the cells from tissue. So the insights that we gain from this model is, first and foremost, why do you need to lot pre-qualify a traditional collagenase product? The best example I can show you is, and this is in a publication by Greer McNally in 2012 on development of a human hepatocyte, a purified enzyme mixture for human hepatocyte isolation, is the variability you see in this type 11. And you can see, in contrast to the original Liberase, there's multiple peaks. And of course, since it isn't purified, it's an enriched collagenase, there's lower amounts, or lower percentage of collagenase in these products. But the interesting thing I want to point out on this is that the collagen degradation activity and neutral protease activity, as measured by Vitacyte's assays, is very variable. Also, you can see has high endotoxin. And you can see that first lot above, the top lot, was the one we used as a model to make a purified enzyme mixture. The next one was a lot that they got but didn't work. And you can see it has high collagen degradation activity, but low protease activity. And the bottom one has just both low protease, collagen degradation, and protease activity. Now, why does this happen? Well, you have to realize how C. histolyticum survives. It has to digest foodstuffs that are in the environment to get the molecules it can use to grow. And so it secretes two different proteases, C. histolyticum neutral protease and clostridium. The real bad actor here is clostridium. It's very hard to separate it from collagenase, even if you're purifying it. And most importantly, it has a very strong ability to degrade native collagen, a native or intact C1, a class 1 collagenase, which is why you see these other peaks. When we made the blend, which I'll share with you shortly, you can see that there's a very wide range of CD activities in 5 different lots of sigma type 11, and the same goes for neutral protease activity and variable endotoxin activity. We made in the purified mixture collagenase MA, which you can see has a very tight CV because it's a purified enzyme, and the same goes for BP proteases, which is used for this application as a very tight CV, lot-to-lot CV. So what do we learn from reverse engineering the type 11 good lot? We first matched the Wunsch activity and protease activity using thermolysin. We find thermolysin didn't really work well. It lowered the viability of human hepatocytes, so we switched to BP protease, which is a DIS-based equivalent enzyme hepatocyte manufacturers. We further optimized this by reducing the collagenase mass by matching on collagen degradation activity, not Wunsch activity. And when we did it, when we tried to adjust the protease activity, we found, interestingly enough, we started 16 milligrams, digest stopped at 8 minutes, way too fast. We cut it in half to 8 milligrams, too long. It's the Goldilocks principles. And around 12 milligrams, it worked just right. So the final formulation was 2,500 CDA units of collagenase MA and 2,200 neutral protease activity units of BP protease per mil of enzyme solution that was used in human hepatocyte isolations. So what happened is you can now, since you know the composition of the enzymes that are required for cell isolation, you can modify these compositions. And this was done at one company who was an early adopter of our human hepatocyte formulation. And what they did is extend its use to rodent hepatocyte isolation. The best of my knowledge, this is probably what they used, about 2,500 CVA units, and they cut back actually the neutral protease activity units. And if you look at Rafe Wittek's seminar that was done in May this year, you can see more of the details, and these details of rat and mouse hepatocyte isolation can be found in his webinar. The third and final insight is development of a rational design model for optimizing collagenase protease enzyme formulations. So, a little bit of history. 2 approaches have been used in the past, and to be blunt, you have to have a company to work with you to take either approach. So I've already shown you reverse engineering of a good lot by Liberase HI, and I just shared with you what we did to develop a human hepatocyte formulation. We've also done this for rodent islets and for human adipose stromal vascular cell isolation, made purified enzyme mixtures for these applications. The second approach is to do a design of experiment. And we did do this, and I'll share the results with you shortly, using intact recombinant class 1 and class 2 collagenase and a fixed dose of BP protease. And really the whole point of the DOE was to assess the effective dose of class 1 and class 2 collagenase on human islet yield and function. The pros and cons of each approach are shown. You can see that in both cases you're using purified enzyme, you're getting rid of contaminants, you should avoid lot selection. The big difference with the DOE is a statistical approach, and the way it's because of statistical design, you can detect interactions between factors. And it's a prospective analysis. You're not basing it on what's been working in the past. You're actually exploring the space, how far you can go. The cons for, of course, reverse engineering is you assume that the good lot is your best mixture, which isn't true. And of course, if you don't have real good characterized enzymes, just because you have a purified enzyme doesn't mean that it's going to be the best enzyme. For the cons for the design of experiments, you need to have tight control of all the variables. You can't have good and bad purified enzymes. You have to know what those enzymes are. You have to adhere to a protocol design. It's really labor intensive. You do a lot of experiments, but you get a lot of knowledge. And again, both cases, you have to have supplier collaboration. So this is the DOE that we did. Is a low and high dose of recombinant class 2 collagenase, either 12 or 20 Wunsch units. Is a low and high dose, as you can see on the top, of recombinant collagenase, either 100,000 CDA units per gram of tissue or 200,000. You can see the pluses are the high and the minuses are the low. And so what we did did a series of isolation. We found in doing this, I should say, when we set up the experiment, we did 3 islet isolations per group using the body tail of the pancreas because that's where most of the islets can be found. The isolators were blinded. They didn't know the collagenase formulation. Neutral protease was fixed. And this work was published by Ballamorgan in 2016. The results are shown on the next slide, which basically in these experiments, they were done at the University of Minnesota, and we paid for them as part of a phase 2 SBIR grant. We controlled the digest to see if maybe we had a bad organ by using NEM, which was a new enzyme mixture that at that time Bala had shown worked better than any other enzyme mixtures that he had tested with recombinant natural purified collagenase enzymes. As you can see, we did a total of 14 isolations and all the isolations worked in all 14 that were performed. And we did, as I mentioned before, 3 isolations for each group, the low, low, high, low, and high. And you can see, much to our surprise, dose didn't make any difference. The important thing to realize is from this study that we're getting about 5,000 islet equivalents per gram of tissue, which if you put it in comparative terms, there was a publication of manufacturing in the CTI-07 clinical trial, the clinical islet transplantation trial. It's been published. Where only 52.5% of the isolations got this amount of islets. The collagenase dose we found, which was a new observation, could be reduced by 25 to 62% when compared to the natural purified. And we also showed for the first time that BP protease was as effective as the C histolyticum neutral protease used for islet isolation. As you can see in the second bar, the NEM body tail that were done at Minnesota and other experiments, it had lower islet yields than what we obtained with the 4 different recombinant enzyme formulations. So further work by Bala, and I'll go through this slide fairly quickly since I want to end on time. But basically what he showed in this, if you took and compared purified natural collagenase at a 60-40 mixture to his low 100,000 CD8 units and 12 Wundt units of recombinant C2, you can see that both are equivalent to 20 Wundt units per gram, as shown on the far bar graphs on the far right side, the orange and blue bar graph. And in fact, it's the yield, when you went down to 12 Wundt units for the natural purified, you lost about half your islet yields. Interestingly enough, is the other aspect that's come out in subsequent studies, and it was also indicated in Bala's early work, that you actually have better function when you use lower amounts of collagenase. Studies now at the University of Edmonton, University of Alberta in Edmonton, have shown that you get significantly better in vitro glucose stimulated insulin release by islets isolated with recombinant enzymes versus natural purified, and you get better recovery, significantly higher recovery of islets after short-term culture, which leads to more islets being transplanted into patients. So that's what we've done in the past. Again, it's something we have to use a company to work with. And as we were revising our website, I realized there had to be a simpler way to do this. And it came to me that we're making this, just like Einstein said, much too complicated. So what I'm going to show you is a model that I've developed with two of the products we manufacture, the PD collagenase products. But this new approach to avoid supplier collaboration is that you just take the elements I've shared with you with this model. You have to realize purified collagenase has really narrow selectivity for collagen and gelatin. It doesn't damage cells. Excess purified collagenase probably has a minimal effect on cell viability. Of course, you can refine it as we've done in the prior experiment I shared with you, but in general, it shouldn't cause a lot of problems. The selection and the dose of protease is the critical enzyme activity that must be controlled to ensure the release of viable functional cells. And to be frank, we're just using, in the traditional collagenase products, we're just using too much neutral protease. We've seen this in a number of feedback we've got from a number of customers. So what are the PD collagenase products? We're labeled in the past, since 2015, DE collagenases, but we simplified and shortened the product line and relabeled them as purified defined collagenase, because literally we showed that these are extremely pure proteins. And you can see we make the PD 100 with 55 in 1 gram of salt dry weight. We put 55 milligrams of purified collagenase, 18 milligrams of purified disbase, and the PD 800. The only difference is the different bulking agent, the peptide, and we use 8 times more collagenase. This material is greater than 95% pure. How can I say this? We actually took all the minor peaks that are in this product and did mass spec on them. They're all collagen breakdown products. About 80% of the collagenase is intact, C1 and C2. The products cover a range of collagenase. The PD 100 is an equivalent activity to that found in 1 gram of Worthington type 1 collagenase. That's the prototypical first collagenase product used back in the 1960s and still used a lot today. The PD 800 composition is equivalent to that found in Liberase HI, has 8 times more collagenase. And of course, as I mentioned, we fix the BP protease activity, and that's equivalent to what would be found in a Worthington type 1 collagenase. So what's important is to get, if you don't have it already, I would highly recommend that you go to these bioprotocol journals or websites, and you can find this information, because what I'm going to be asking you in the first step of the procedure is to determine your mass of collagenase. Ergo, you don't have to consult with VitaCite. You just have to find out about how much collagenase you're using in your current procedure. So step 1 is determine the mass of collagenase in the product. Roche Libraries Research Products tells you what that mass is. Worthington types 1 through 4, they bottle based on collagen degradation activity. So all these products will have about the same mass of collagenase. So for Worthington type 4, it contains about 6% collagenase by weight, so you divide by 16.67 the dry weight that's in the bottle, and you figure out how much collagenase is in the bottle. Note that Worthington, a large part of Worthington's business, private labels, so other suppliers could be using the same products. For other products, you'd need to contact VitaCite Technical Support for guidance, because the Sigma products are extremely variable in compositions. In step 2, you got to calculate the mass of collagenase. So that's just a simple thing. How much is the bottle? How much you're putting in the enzyme solution? And you need to know the mass of collagenase in the enzyme solution. In step 3, you're going to prepare 3 solutions of PD, a PD collagenase 100, an 800, and a 50-50 mixture of the two. To illustrate how you use this approach, we'll use the example. Well, first and foremost, you'll see these all have different percentage of collagenase. Apologize. And to use this approach, what you're going to do is assume we're making a 10 milligram collagenase solution in 100 ml, or 10 milligrams collagenase in a 100 ml solution. You're going to divide 10 milligrams for the PD 100 by the percentage of collagenase, and to get 10 milligrams, you're going to have to use 181.8 milligrams of the PD 100, and so forth, all the way down the line. So if you make these solutions up, again, this illustration of a 10 milligram collagenase 100 ml solution, you're going to find that you can make a fixed amount of collagenase, as shown on the top here, in 10 milligrams, but you're going to have variable protease activity. So as you can see, this would be an equivalent for the PD 100, what you'd find in a type 1 Worthington type 1 collagenase, and you'd get about a fourth of that, about a half of that in the 800. But the bottom line is, what you're in essence doing is saying, okay, as long as I get a good amount of collagenase that's in excess, what I really want to look at is the protease activity. And this model actually accomplishes that for you. So step 4, of course, would be, does the formulation work? And you have to note, as you decrease the protease activity, as I showed you in the human hepatocyte, you're going to slow the digestion down. So you're going to have to balance the activity with functional cell recovery in the time of digest. If it doesn't work, and we've found this, you may have to supplement the enzyme mixture with purified clostridium. And I'm not going to get into this today, but you can see our clostridium pack insert for details. The reason you may have to add clostridium is it has a different selectivity than the BP protease. It's a trypsin-like protease. And using complementary enzymes may provide better cell isolation results. This is extremely user-dependent. So if you compare your traditional lot qualification versus the optimization, you see that the amount of effort required is similar. The knowledge that you gain from this, you're gaining no new knowledge from a lot qualification. But in essence, you're defining the enzyme composition when you do the PD collagenase optimization or any other purified enzyme composition. You don't get around pre-qualifying lots in the traditional method, but once you define an optimal formulation, you don't have to pre-qualify new lots. It's obvious you can't, you can modify the formulations with purified enzymes. And of course, we have good data based on collagen degradation activity that it's good for 4 years. And of course, at times, as I mentioned before, for the collagenase P, you don't know how stable it is over time, because more than likely they're using a peptide assay to measure it, and that doesn't correlate. So I want to end on this one note. Why is this all relevant? It's all relevant because there was a new standard published for insulator materials, the 20399 standard, and the supplier's responsibility is to consistently manufacture products. And the important thing is so that you get consistent performance. The AM user, the insulator material user, controls the insulator materials by taking a risk-based approach. What's a bigger risk than collagenase that affect everything you do in any enzyme-mediated cell isolation procedure? And in fact, the relationship between the user and the supplier should be cooperative and transparent to ensure performance of the products. I won't spend a lot of time on this slide other than to say that there should be an interaction if you really are developing, especially a clinical procedure to isolate cells. And the important thing for the supplier is what I've shown you in this presentation is to identify all the molecular components and why it's important to have certain characteristics of your collagenase. They have a reproducible ancillary collagenase, ancillary material. So our goal is, in essence, we found ourselves in the story transforming tissue dissociation. So obviously, why I'm here today is to improve and share knowledge of tissue dissociation. We manufacture consistent products and develop cost-effective products to meet needs. And really what we want to do is increase your productivity. So in the end, if, as W. Edward Deming said, the father of quality in Japan, basically, it's all about reducing variation. So, thank you for your attendance. And I'll see if we have any questions.
So then when you go to like market protocol, you just pick the one that is.
Molly, you have any questions?
Six.
Oh, yes, Bob. I hope you can hear me.
Yes, I can.
Okay. No, it's a very interesting presentation, Bob. If you compare enzymatic activity between PD800 and Vitacyte HA and recombinant collagenase.
Sure.
Do you see any difference? Like I'm trying to understand the PD800 or D800.
The PD800 has a different class 1, class 2 ratio. It has about, which is typical of most collagenase products. It's a 75% class 1, 25% class 2. As you know, the natural purified is a 60-40, 60% class 1, 40% class 2. I think the activity of the recombinant is about a 50-50 mixture, but they're all about the same that we sell. That doesn't say that the experiments you did, to my knowledge, and I'd have to go back and look at it since I haven't thought about it, you're using much lower amounts of collagenase degradation activity, because the low was 30% class 1 and 70% class 2. But that was the only exception. But see, we don't sell that. We sell in the middle. The product we're selling, our collagenase HI is about a 50-50 mixture, because it's in the midpoint of all of those tests.
Sure, Bob. The reason I'm asking this, when I try to use 12 Wunsch unit per gram pancreas with the Vitacyte HA product, definitely that is an insufficient dose. 12 Wunsch unit is really low dose. But at the same time, with the recombinant enzyme, I can digest the pancreas.
Yes.
Similarly, if I use 12 Wunsch unit of PD800, of course, there is more CDA activity in it. I don't know whether the 12 Wunsch unit will behave similar to Vitacyte HA product or recombinant enzyme product.
That's a good question. I don't know the answer.
Okay. Maybe I can try to isolate.
Yeah, I don't know the answer.
And one more thing, Bob, for your slides are very nice to see the comparison between one product versus another product and all. Similarly, I would like to see 3 different proteases, thermolysin, BP protease, and the neutral protease. If someone wants to really compare, chemical aspect, not biological activity in general, what type of difference we can see? Because if we have a slide like that, it's easier to explain to investigators. These are the differences between 3 different proteases for them to easily select.
So you were mentioning thermolysin, BP, and the C histolyticum neutral protease, right?
Exactly.
The interesting thing about this is where it gets very complex. And why it gets complex is because all 3 of those enzymes, to the best of my knowledge, are in the M4 protease family. They all will degrade a defined, or they all have within, let me rephrase that. They all within their structure the same residues involved in the peptide cutting. All of them are the same. That's why they're in the same family. The problem you get into is when you get into natural substrates. If you use I can tell you there's a paper on this. If you use thermolysin and disbase or BP protease to strip cells off of amniotic membranes and prepare, a biomaterial in the amniotic membrane free of cells, I can tell you the thermolysin will give a very smooth surface. The BP, the disbase will give you a bumpy surface. And yet these are all the same. So it's very difficult for us, and we've talked about this, I know, in other conversations, to know how to explain it, because it's all empirical. And I think that, let's face it, in your prior unpublished work, you have data which indicates some difference between these enzymes. But there's very little knowledge about it. And I guess what we're trying to say is, how do I want to put it? Really, the focus should be on exploring the neutral protease. I think we've solved the collagenase problem.
Yeah. think Dr. Stu has two questions in the chat box.
Okay, I'm sorry.
Stu Williams says, great talk, Bob. You provided a 4-year stability based on CDA. Do you have corresponding functional stability, i.e. cell yield?
No, we all do biochemical assays. Everything we do is biochemical assay. I don't think that's an interesting question, whereas the people on the other side, i.e. those who use the enzymes, do cell yield, but we don't do any cell yields here. And I think the other issue, just to bring out, is, of course, we don't know how broad the range is for cell yield. It's going to be dependent on different types of cells. And I guess the best example I can give is you can have some people use virtually any collagenase and always get cells because they don't need a lot of them. Whereas others like Camillo Ricordi, who needed as many islets as he could get, had to come to Beringer and say, can you help me? So.
And then where are we regarding standardized C of A for enzymes used in tissue digestion? Is CDA becoming a standard?
That's a very good question, and I can tell you that I tried to get our kinetic fluorescent microtiter plate assay adopted by U.S. Pharmacopoeia, and they decided not to. We actually have an alternative colorimetric assay that we'll hopefully begin working on in Q1 2025. And the goal of that is to develop a standard assay, because that's the big gap in collagenase characterization, is we're measuring the wrong activities. And so that's going to be work ongoing, and we're hopefully going to get some intern from the local biotech programs to come and work with us for a quarter to get that assay up and running. It's already working. My colleague Francis Dwelleth is using this very simple assay, colorimetric assay, routinely. And now the question is can we make it a transferable assay?
Okay, and last question from Stuart is optimization of enzyme utilities, good organs. Any thoughts on how to optimize enzymes for bad organs, also defined as the organs we use in transplants?
Another good question. I can tell you, Greg Zott once told me when they got a bloody pancreas and tried to get islets out and the islet isolation didn't work, and this is, of course, in a special circumstance where you can't get all the blood out of the organ after flushing. What he ended up doing was just injecting a whole nother dose of enzyme into the organ, and it worked. Don't know any more details than that. And of course, Bala, who's on the line here, could tell you every chronic pancreatitis organ is a bad organ. You have to take extreme circumstances to get the islets out on some of them. So This really gets down to, and this is why it's so important when I've talked about this before, the three important factors for any cell isolation procedure, especially human tissue isolation, is you have to have a reasonable organ. You have to have a very experienced team. They know what they're doing. You can adapt based on it. And you have to have the right enzyme. And what I keep on saying is the only thing that you really can't control unambiguously is the enzyme. i.e. why we're trying to transform tissue dissociation.
Yeah, I can add a few points for Dr. Stu's question.
Okay.
For example, the bad organs for allotransplantation related from brain dead donor. So maybe we can categorize the pancreases, bad pancreases, into two categories. One is maybe pancreas comes with longer cold ischemia we know that it's going to give low islet yield. And if the pancreas from lean donor, evidently it is going to give a low islet number. So I would say, those are the two bad organs. I think adjusting the enzyme dose is the best way to get maximum number of islets from those organs. For example, if cold ischemia is more than 10 hours, 12 hours is kind of our cutoff. Then in that scenario, generally we try to reduce the protease dose. That really helps to get, instead of using just a standard one vial approach of collagenase and protease, reducing the protease helps in those scenarios. And for low BMI donors, high BMI donors, no matter what we do, whatever the enzyme dose we use, generally we get a very good number of islets. Low BMI pancreases are a problem. For that, again, just increasing the protease dose is really necessary to get a nicely cleaved islets. It's almost similar to our young donor protocol, reducing the collagenase, increasing the protease for those lean donors. We see some better quality islets or good number of islets that we see. For chronic pancreatitis cases, again, we kind of separate into two different categories. It's all about the severely fibrotic pancreas. Mild and moderate case, we can easily manage without any problem. The severely fibrotic case is the main problem. For severely fibrotic cases, if we know we are not going to purify the islets, that means we can really reduce the protease dose. Even if the islet, because if we reduce the protease dose, islets will be embedded. That's okay because we are not getting large tissue pellet. It's easier to infuse. But at the same time, if the pancreas is very large, we are going to get large amount of pellet. Evidently, we have to increase the protease. To kind of summarize, I think adjusting the dose based on the nature of the pancreas, whether it is from a pediatric donor, older donor, lean donor, long cold ischemic organ, adjusting the dose will help.
Okay. So I think we're at the bewitching hour. So I hope you enjoyed it. I was, as I was writing this presentation, I realized to myself we had a celebration in 1994 where Ray Rajat and Camilla Ricordi came to Indianapolis and we celebrated the launch of Liberase. And again, that was November 28th, 1994. So I hope this seminar or webinar showed you how far we've come. Still slow progress as the issue that you brought up with the proteases, but I think if we can stop fiddling around with the collagenase and realize we have that solved and start asking questions about the protease, we'll be a lot farther along and we'll make faster progress.
I think we learned a lot and we evolved, plus I think your contribution is really grateful, Bob. Now
it's like everyone else, we all try, right? It's all you can do. So thank you for attending. All that are there. Thanks, Stu, for the questions, and have a good, happy Thanksgiving.